Okay, hello again. We are resuming our seminar, and we're now starting the fourth and last session of the day. And the first speaker will be Diriba Nemera from Ethiopia. Diriba, please.
Good afternoon, everybody. I'm Diriba Nemera from Ethiopia. I'm going to present the hydraulic parameters of citrus trees irrigated with secondary treated wastewater. And the research was conducted in, under the supervision of Dr. Shabtai Cohen in Volcanic Research Institute. Citrus is one of the major commercial crops grown in Israel, as well as in tropical and subtropical areas throughout the world, for it is economical part of the fruit, and it is grown under intensified agriculture system where it needs inputs to give good quality of products, and it requires large amount of water because the fruit consists of the majority part is water, which consists around 90%, and in addition it needs more water for the synthesis to be produced in the leaf and for the translocation of synthesis that can be produced in the leaf to the fruit. And in addition, it produces wider canopy that can transpire more water, and the crop needs, and it will be dependent on the irrigation water, especially in dry, arid, and semi-arid areas where the rainfall is erratic. However, the fresh water for irrigation is becoming a serious issue in the world because of high competition on the fresh water for the domestic use, because of the global warming that causes erratic rainfall and degradation of natural resources that can contribute for the management and the wise use of water. And we can see in different parts of the world, people are suffering for water, even for drinking. As we can see from this, these people may be dependent on this water for its domestic use. And in this picture, we can see how many pupils are competing for water that can be provided from the municipalities. Surprisingly, there is a forecast that in 2015, there will be a serious water crisis for irrigation. That means water will be the major factor limiting the productivity of agricultural crops that may put the agricultural crops production under pressure. So if water is becoming the serious issue in the near future and still now, what should be the solution? We are interested to study the solution for this problem. And the researchers are encouraged to study what is the effect of this treated wastewater on the crops, and whether it can be used efficiently for the production of agricultural crops without harming problem on the crops. And this treated wastewater seems to be important for crops because it can alleviate the competition on fresh water. As the population is growing, the amount of water that we can use for domestic will be increasing, and we can get sufficient amount of water to irrigate if we treat it in a proper way. And the water, the treated wastewater supply will be constant, and it is cheaper. I can show you in evidence on the next slide. However, this treated wastewater should be used in environmentally friendly way because it may cause environmental pollution if it's not appropriately used. Here, this picture shows that water use in Israelis. As we can observe from here, majority of the water is used for irrigation in 2003 according to this report. And when we see the trends in the future, in 2015, it is supposed the amount of water that's required for domestic will be increased by 200%, while that of the agricultural demand is not increasing this much, even though there is a trend. So this needs to find an alternative water source for irrigation. And in addition, this treated wastewater is cheaper as compared to the fresh water. for the farmers, it seems reasonable to use this treated wastewater for irrigation. As we can see from this graph, the cost of treated wastewater per meter cube is around 0.6 from this, and the freshwater is around 1.5. That's a great difference in terms of cost for the grower to use this water for producing agricultural crops. Generally, in Israel, the amount of wastewater used for agriculture accounts about 70%. From the total amount of water used or recycled water, 70% is used for agriculture. And the overall water withdrawal or water use in Israel is dependent on marginal waters, which means only 47% of the water use in Israel is dependent on the fresh water that can be obtained. Based on this, we hypothesized that secondary treated wastewater will have a detrimental effect on the hydraulic parameters of citrus trees. And we set objectives to achieve our hypothesis. And our objective was to study whether the secondary treated wastewater may have a detrimental effect on the citrus tree hydraulic conductance, the canopy conductance, and thereby to estimate the effect of climatic factors on the tree hydraulic conductance and the canopy conductance that can contribute for the overall growth and the development of the plant. Materials and the method. And the study was conducted in a citrus orchard at Mizra, Israel. And the experimental treatments were secondary treated wastewater, as we can see from the design of the plot. The blue one is representing the secondary treated wastewater, which was replicated 6 times, and as well the control was replicated 6 times. And you can see here other 2 treatments that was supposed to be implemented in the future. Hence, the project is a multidisciplinary project that included different disciplines to study the long-term effect of treated wastewater on the overall performance of the crop. And there were measurements that was made to achieve our objectives. The first one is we measured the sap flow per tree by using the thermal dissipation probe that were developed by the Granner, and we computed the sap flows from the temperature difference. In this method, 2 thermal dissipation probes were inserted into the stem of the plants, into the xylem, and the upper probe is constantly heated by providing 0.2 watts power to heat this probe, and the bottom is not heated. In this case, when the water moves from the soil into the plant through the stem, the heated probe will be cooled because of the water fluxes, and that can be done through the heat conduction through the probes. And the thermal dissipation probe read this temperature difference, and the data logger will read the temperature difference in between the 2 probes. And from this temperature difference, the sap flow per tree were computed by the formula I showed you before. The other thing we measured was the leaf water potential, which was done by using the pressure chamber. And we selected the fully grown leaves, and the leaf was cut, and we put it under the pressure chamber. And the pressure chamber was, the leaf was put in the pressure chamber, and we applied the pressure through by controlling this. And when the positive pressure applied on the leaf in the pressure chamber will become equal with the negative leaf water potential in the leaf, the water droplets come out, and we observe by the hand lens, and we record the pressure at which the water droplets come out of the leaf. That is then leaf water potential at that time. And we used this water potential for the computation of sap flow. Hence, I will not present what the water potential was here. Hence, we used it for the computation of hydraulic conductance. The hydraulic conductance, which is the ratio of sap flux density and the water potential gradient, was computed by the formula that was developed by Cohen and Naur 2000. And from this hydraulic conductance, we computed the hydraulic resistance. That means by how much the different components or the path of water flows from the soil into the atmosphere will pass through different hydraulic conductance, which is the inverse of hydraulic conductance. And the other parameter computed was canopy conductance, which is a function of sap flow, radiation, humidity, and the air temperature, including the wind, that can influence by how much the canopy of the plant is conductance for different the carbon dioxide that the plant needs, and the overall canopy conductance of the plant was computed. The data was analyzed by using the JAM software. Results and discussion. The first interesting result we found from this experiment was there was higher hydraulic resistances in the citrus trees irrigated with secondary treated wastewater. Especially, there was highly significant hydraulic resistances from the soil into the stem than the stem to the leaves. And in this case, we are also interested to see where this significance was intensified through the course of the time. And here is the series path of hydraulic conductance from the soil into the atmosphere through the plants. And we found here the hydraulic resistance found in the soil, in the root, in the stem, and in the leaf. And when it goes into the atmosphere also may have the resistance for hydraulic conductance, so that if there is strong hydraulic conductance in the root, the plant may not get sufficient amount of water. That was observed in the first result. Is under secondary treated water, we found high resistance for the hydraulic conductance. Based on this, we are interested to see at which time of the day this hydraulic resistance prevented the flow of saps in the plant. And we found here from the middle of the night until 2 a.m., there was no difference in the sap flow in both treatments. But afterward, we found the higher sap flow in case of fresh water, whereas it's reduced in case of secondary treated wastewater. And normally in both cases, it seems that the highest sap flow was observed in the middle of the day. That is interesting as to see if this hydraulic the flow of saps may contribute for the overall performance of the plants. And we checked for the hydraulic conductance, that means through the sap flow, if it is influencing the overall the day or if it is specific to some part of the day. And here, the average of the sap flow per tree in the 24-hour averages, including the overnight. And the maximum sap flow was observed in fresh water, which is significantly different from secondary treated wastewater. Whereas in the midday, there was highly significant difference in sap flows in fresh water, which is more higher than that of the overall average sap flow. This is because in the nighttime there is less sap flow that may cause lower sap in case of the 24-hour average. And by observing these results, we are interested to see what are the other contributing factors for the variation in the sap flow through the plant. And we have checked and estimated the effect of air vapor pressure deficit, which was manifested as the atmospheric dryness. How the atmospheric dryness or the dryness of the atmosphere will influence the sap flow in plants. And we found that there is no as much relation between the sap flow and the vapor pressure deficit. When the air became drier and the higher vapor pressure deficit, we found less subflow as compared to where the vapor pressure deficit was lower. And this may tell us the finding shows us the vapor pressure influenced more the subflow in secondary treated wastewater, which was reduced than the fresh water. And we are also interested to see from this picture, it seems there is no much difference. The effect of vapor pressure doesn't show more difference. And we are interested to see if other parameters or the climatic factors can influence this subflow. And we estimated the effect of net radiation on the subflow and the reference evapotranspiration, which is the contribution of the climatic factors around the crop. And we found on this result, there was a highly correlation with the net radiation and the evapotranspiration around the crops. Whereas the water movement in the plants, that may be in the form of subflow, is less influenced with the increase in the net radiation that we can observe. However, in between the two treatments, the water flow in the plant is more reduced in case when it's irrigated with secondary treated wastewater than when it was irrigated with fresh water. And the other interesting thing we found is here the effect of vapor pressure deficit on the canopy conductance. How the vapor pressure can influence the canopy conductance, because we consider this canopy conductance is very important for the plant, for its growth and development. And from this result, we found that there was a negatively correlation between the canopy conductance and the vapor pressure deficits. As the vapor pressure deficit increased, the subflow also decreased. That is causing the plant may not respond in accordance to this vapor pressure deficit. And this may be due to the ability of the plants to manage its canopy, especially the stomata. In the mild vapor pressure deficit, the plant will open the stomata to take in carbon dioxide that can be used for the assimilate production and for the plant growth. At that time, the plant will lose water and it gains carbon dioxide at the expense of water. When the water becomes shorter, the plant will close the stomata and there will be lower subflow as we can observe, and there will be less canopy conductance when the plant closes the stomata due to the lower water in the plant, and when the atmosphere becomes more dry. That is the reason we can find here the canopy conductance is decreasing as the vapor pressure deficit is increasing. The last result we found is the leaf area specific hydraulic conductance. We found that in the first result I presented, there was highly significant hydraulic resistance from the soil to the stem, which influenced here the hydraulic conductance from the leaf. In the leaf is lower from the soil to the stem, and from soil to the leaf is lower. But we find here the highest subflow in both cases. Especially the highest subflow was observed, or the hydraulic conductance was highest in case of the trees irrigated with fresh water as compared to the secondary treated wastewater. From this experiment, we can conclude that secondary treated wastewater significantly reduced the subflow and the leaf hydraulic conductance in the citrus trees, according to this experiment. And the hydraulic resistance was increased from the soil into the stem as compared to the other part of the plant, and it was higher under the secondary treated wastewater. And we conclude that canopy conductance was influenced by the climatic factors, in particular the atmospheric dryness that reduced the hydraulic conductance in a large amount, as we have seen, and it's leading to the stable subflow in case where the atmosphere is becoming more dry. However, as this project is ongoing, and the crop is a perennial crop, that the effect may be manifested in the long time, it is the recommendation of using this secondary treated wastewater as an alternative use for irrigation should be recommended after getting the final results that was ongoing in the project. And the other factor that may be the yield, quality, and the plant health should be also addressed under this study to get the final result or the conclusion of using this wastewater as an alternative for citrus production. And I want to address my grateful thanks for the PEARS Foundation who that sponsored our study, as well as the Hebrew University, Volcano Research Institute, Dr. Shabtai Cohen, and also this all who contributed in one or another direction for our study and for the finalization of this research activity. Thank you.
Thank you very much, Diriba. Let's start with the questions. Gabriel.
What I don't understand is why, in the first place, you started with the investigation about the wastewater, with the assumption that wastewater may cause detrimental effects on the trees. In this case, like what kind of compounds or what provoke this effect on the trees?
Okay, if I get your question is why we are interested to study the effect of secondary treated wastewater on plant performance, or
like what led the investigation to investigate the research to investigate this? I mean, what does the wastewater has that could affect negatively the performance of the tree?
Okay, the intention was, as you know, the wastewater is known to have many impurities as compared to the fresh water that we can get from the underground. So that we are interested to see whether these impurities, that maybe when we compare the fresh water with the secondary treated wastewater, we can find the higher salt content. Maybe there are some pathogens that is impure, hence we are not going in detail of the processing that it can be used for the drinking purpose, for example, so that many things may be found in that case that may influence the overall water uptake in the plant or the plant health. So that the intention of the project is not only to this, as I said, described in the methodology, it is a long-term project. Even there is a study on whether it harms the soil structure, maybe the soil characters, and also the overall the crop performance.
Do you want to ask a question?
In sap flow measurement.
Okay. Sap flow measurement.
The picture.
Okay.
Yeah. So you have written heated and unheated. So can you explain like that's the unheated? I mean, you have put the arrow. So how it is like?
Okay. In this case, two probes are inserted in the stem or into the xylem of the tree. The two probes were connected into the data logger. Here, in this case, we expect the sap is flowing from the soil through the stem. Because of the water potential gradient, we expect the water to move when the plant needs. In this case, this probe is not heated and this one is heated. Constant heat is supplied to this probe. And when the sap flow, the temperature at this probe is reduced through the heat conduction system, that the heat can be cooled. And at this time, we can measure the temperature difference between the two probes. In addition, for example, in the daytime, we expect high water movement or sap flows. In that case, you will read less temperature difference. However, in the nighttime, you will get more temperature at the two probes, or the difference between increases. That means the sap flow is lower.
Daniel?
Two questions. First, I'll ask on this one. At what point, where is the temperature measured on this scheme?
Where?
Where is the difference in temperature measured?
Here, the probe is inserted here. Maybe the distance between these two probes is 10 to 15 centimeter. And the data logger reads the temperature difference that can be found between this and this. This is not heated, no heat source here, and it is lower in temperature. And this one is constantly heated by 0.2 watt. It reads the temperature.
Right. So it's measuring the sap temperature flow?
The sap flow in the xylem.
Not, okay.
In the xylem of the
Is
tree.
there a separate probe that measures the temperature at the heated probe?
Okay, the two probes are connected into the data logger that can interpret the result between the temperature difference between this and these two probes. And it gives only the temperature difference between this. Because the temperature or the heat is provided 0.2 watt from the power source there, and it's recorded this. And when the sap flows, this temperature will be reduced. The difference in between this temperature is due to the flow of sap from the soil into the steam.
Okay, my second question is, if you can tell me some of the background, what qualities of the secondary treated wastewater actually contribute to the difference in the hydraulic conductance and in the sap flow? What components of the wastewater lead to that difference?
Okay. In the wastewater, as compared to the fresh water, there are many differences. Among these, the first one is the total suspended solids that we cannot find mostly in fresh water, but they are found in a higher amount in secondary treated wastewater. The other thing that may influence the secondary treated wastewater is the biological organisms that demands on oxygen. Unless they get oxygen, they cannot decompose, and they may affect the sap flow. Due to this thing, we may expect some things that may happen in the aquaporins that may take water from the roots. They may be clogged, or there may be any other factor that can hinder the uptake of the water from the soil into the root and the other way of process through the plants.
Okay, so
No, Shilpa first, please. And I'm afraid this will be the last question. Sorry.
My question is related to Danny's question. Like before you're using the secondary wastewater for experiment, are you going to check any properties of the wastewater like electric conductivity or nutrient content, nutrients available in the water or solids or something like that before you're going to carry the experiment?
About the fresh water?
Fresh water and recycled water.
To compare between the two treatments. Yeah.
Compare the qualities of the water used in the two treatments. Is this what you mean?
Yeah.
Yes.
Are you going to check before you're starting the experiment?
Yeah, it was checked, the difference between the secondary treated water. Because, for example, in case of fresh water, mostly the total salt content that can be expressed as the decimal is less than 2. But in case of the secondary treated wastewater, it is more than 2 decimal. And the other thing, as I explained in the other components, for example, the SAR, that means the sodium absorption ratio, that is the ratio of sodium to calcium is higher in case of the secondary treated wastewater as compared to the fresh water that can influence the water uptake.
So there may be a difference in electric conductivity. of
Right, right.
Okay, thank you very much. And we shall go on to the next speaker, Mr. Fatta Gurung from Nepal. Sorry.
No need to put here. No need to put here. Good afternoon, everyone. I am Fatta Gurung from Nepal. Today I am going to present my presentation on my topic, effect of compost on soil fertility and nutrient availability under the guidelines of Dr. S.R. Bartal from Volcanic Research Center. Before I start my presentation, I would like to present some glimpse of research problem. In Mediterranean, the duplication of soil organic matter is a big problem due to the intensive farming system. It lead to reduce the productivity of the soil due to low nutrient cycling, degraded physical stability, and high incidence of soil-borne disease. As we know, compost is very popularly throughout the world for organic farming. What is compost? Compost is made up of by the decomposition of natural organic wastes like plant residues, animal manures, through the action of microbial activities. It also provides the good source of organic matter with accessible nutrient, micronutrient, macronutrients, and moreover, it also improves the soil structure, water holding capacity, soil microbial activity, nutrient supply, and enhance the root development. We hypothesize that compost is expected to improve soil nutrient availability and soil fertility due to its contribute to increase soil organic matter and soil microbial activities. But compost may have negative effect due to enhanced soil salinity. And our objective is to investigate the effect of the compost on soil fertility and also to check the effect of the applied compost on soil chemical properties such as soil organic carbon and organic nitrogen content, available nitrogen, phosphorus, potassium, and soil salinity. Material and methods. The research was conducted on the Nemera research station, which is located on the northern part of the Israel. There was 4 treatment, organic, which was divided into the 3 dose of the compost, 20, 40, and 60 meter cube per hectare in the volume, and another is manure fertilizer, which is act as a control. We apply 0.3 meter cube per hectare. This is our field experiment. The treatment were organized. Each treatment organized in 5 randomized block design, and here length of the plot was 25 meter and width 18 meter. The soil sample were taken from the plot at the flowing depth 0 to 5, 5 to 50, 15 to 30, and 30 to 60 centimeter. We divide the soil into different layers because we also want to see the leaching ability of the nutrient. And from the soil, we measure the flowing variable in the lab, available ammonium and nitrate, which is extracted by potassium chloride, available phosphorus, which is extracted by bicarbonate, and soluble potassium, organic carbon, and nitrogen in the soil, and other electric conductivity and chlorine. before I am going to present the result, I want to explain about the inorganic nitrogen and organic nitrogen. Inorganic nitrogen is generally three forms: ammonium and nitrate and nitrite. Nitrate are generally small amounts, so we can't measure. We are not measuring. And organic carbon, which are incorporate in the living biomass, such as protein and amino acid. And results: organic carbon and nitrogen. I'm going to focus just the result of the October 2012, which I did. Here is the percentage of the carbon significantly higher in compost than the control, and organic nitrogen, which is also higher in compost than the control at the depth 0 to 5 centimeter, and organic carbon and nitrogen at depth 5 to 15 centimeter. We also found the higher amount of carbon and nitrogen on the compost than the control treatment. And inorganic nitrogen, ammonium concentration, there was no significant effect between the control treatment and control, but we found that the amount of concentration of the ammonium decreased with increase the depth of the soil. It's due to the slowly leaching of the ammonium. And nitrate concentration, there was also not significant effect between control and compost treatment, and but higher amount of ammonium concentration was found in the top layer, but after the 10 centimeter depth it was no effect of the treatment. And available phosphorus, there was significantly higher in the compost than the control, and also we found that we increased the dose of the compost, there was higher level of phosphorus on the soil, and potassium also we found significant effect between the treatment beyond the 10 centimeter depth. There was no significant effect on the potassium concentration. And chlorine and electric conductivity, which measure the salinity of the soil. Both electric conductivity and chlorine was significantly higher on the compost treatment than in the control in the top layer of the soil. I summarize all the results that organic carbon and nitrogen at the compost treatment significantly higher than the control. It shows that organic carbon and nitrogen very important for long-term maintenance the soil fertility. And inorganic nitrogen like nitrate and ammonium, there was no significant effect between the control and compost treatment. This result just reflect that nitrogen availability for the plant, and it will be downward leaching at the specific time. And available phosphorus and potassium significantly higher in the compost treatment at the top layer, because phosphorus PN ratio is higher in the compost, therefore higher concentration of the phosphorus we found in the compost treatment. And soluble salt like chlorine and electric conductivity significantly higher in the compost treatment than control. This result show that compost application had modest impact on soil salinity, and thus overall finally we conclude that in the long term compost proved to be best solution for restoring soil fertility with organic carbon and nitrogen. And our future work combine the compost with the biochar, because it will have the synergetic effect on soil fertility and productivity by retaining of soil nutrient, increasing soil organic matter, and enhance soil microbial activity. I want to thank to my mentor Esser Bartal and all my lab members who helped for in research, and all of you for attending my presentation. Thank you.
Thank you. Questions, please. Yes, Daniel.
But I was wondering if you could, well, first question is, are there any ways to compensate for the high levels of salinity from the compost?
Pardon?
Are there any ways that you know of that are being used to compensate for the higher levels of salinity from the compost use?
Yeah, it's their compost salinity is a certain level. If there was electric conductivity higher than the 4, then it will be salinity. But the compost is not much increase the salinity of the soil. But another alternative, we can use biochar, which will maintain the salinity of the soil.
What is this, the biochar?
Biochar, which is made from the burning of the plant material, and there is limited amount of the oxygen, and it's a new technology, and now it increases the water holding capacity, and also it conserves the carbon. It conserves the carbon to the soil.
You probably mentioned this, but what were the control plots given?
This experiment?
Yeah.
Yeah, mineral fertilizer.
What?
Mineral fertilizer.
Which one?
Urea.
Only urea.
Yeah.
So, so no surprise that potassium and phosphorus were lower under the control.
In control
to the compost,
compost
treatments.
Because in mineral fertilizer there was a less amount of phosphorus and a higher amount of nitrogen.
Urea is only nitrogen.
In mineral.
Potassium and phosphate are applied as a regular practice in agriculture. So it's long-term experiment. So it's dependent cycle of crops. So if specific crop require potassium or phosphate, is applied according to the demand, to the requirement of the plant. While as Fatta said, the ratio of phosphate to nitrogen is relatively higher in the compost than is the practice in mineral fertilizers.
If Asher already mentioned the long term, what do you know about the possible long-term effect of applying compost to such plots? If you were continuing this experiment, let's say, following the same plot for the next 5 years, what would you expect? How far would this effect go?
The compost application, it like mineral fertilizer, just take the plant in short time, but application of the compost because it take a long time to the degradation, and also there is increase the microbial activity. That's why it's conserve the soil fertility like carbon and nitrogen for a long time.
What do you mean by long time?
How much?
6 months, 1 year, 10 years?
No, it's
Of course, it depends on weather and other conditions, but roughly, what time scale?
it's 1 year, 6 months, 1 year.
So 1 year, so that means that for the next cropping season, there will be no impact of the...
Not will be completely decreased, but it will be decreased in time to time, but not certainly it will be not decreased like mineral.
So if you would be asked to recommend a farmer how often should he apply compost to his field, should he apply every year, every 2 years, 3, 4? What would you say, in order to maintain good fertility of the soil?
Yeah, it's every year.
Every year?
Yeah, every year.
Other questions? Shabtai, please.
Yes. Do you know what the desirable levels of nitrogen and phosphorus would be relative to what you found there?
Desirable amount?
What amounts? If you can go back to the table of what you found. Are there desirable levels? I don't know. Miri said this. And undesirable
Exactly.
levels.
What are the best levels? What are the best levels for the plant?
For nitrogen?
Recommended.
It's a table. Go back to the table of statements.
Like this?
Okay, maybe it's the wrong question.
So,
I assume that organic carbon, it's better to have as much as you can, right?
Yeah.
But with nitrogen and phosphorus, perhaps there's an optimum level.
Yeah, but I don't know that exact level.
okay, other question? Rabi,
You're trying to state the effects of compost on soil properties.
Yeah.
What soil properties did you measure, and if you did measure any?
Soil property?
Yeah. Try to state the effect of compost on soil.
Soil fertility.
Okay, your abstract is different from what you're speaking of. So, okay, what's the effect of this your compost application that you did? Now, what's the effect on the soil properties in general?
Yeah, it's increase the organic carbon and nitrogen for the it increase the organic carbon and nitrogen, and also
Any effect on
yeah,
texture,
phosphorus.
structure,
Yeah, I didn't measure.
texture, hydraulic conductivity of the soil.
I didn't measure. I measured just.
Not what you measure, what you may know from the literature.
Yeah.
Do
It's
you
been
have any idea?
Yeah, it increased the soil. What is it? Soil texture, soil water holding capacity, and hydraulic conductivity.
Do you think
Just a second, I had Philip on my list.
Do you know in what extent how much is it used as compared to other organic fertilizers? And do you know how costly compost is? Like, for example,
Yeah, it is costly than the mineral fertilizer. Yeah, it's cost. But I don't know how much. It is costly than the mineral fertilizer.
But is it used more than other organic fertilizers?
Other organics mean? This is organic. I mentioned about the organic compost.
Is compost like a routine
Yes,
treatment?
I mean compared to other organic fertilizers, like is it like most widely used or?
Yeah, it is most widely. Not like also we can make the compost from the what to say residues of the oil extraction. After the oil cake, but it's made of by the plant residues and other wastage.
do you think constant application of a compost could have a negative impact on the plant physiologically?
Yeah, if we apply the compost regularly, it's increase the there was a higher concentration of the phosphorus, and then it will negative effect to the plant, because if a plant will take the more amount of the phosphorus, then affect the other nutrients like potassium and chlorine, others, it affects to the other nutrients.
Okay, you said that the application of compost will minimize the leaching of nitrogen. Concluded at the end. Under the compost treatment, you found higher nitrogen. That is probably due to the prevention of leaching, you said, I think. Or if not, how composite will contribute for the increasement of available nutrients as compared to inorganic fertilizer?
For leaching? Leaching mechanism?
How it prevents?
For the leaching?
Yeah. There may be anything related to the improvement of the soil structure or something.
Yeah, we apply
how to contribute
it. We applied to the compost for the leaching. We found that higher leaching in the inorganic nitrogen, but in the compost there was not much leaching in the compost. So we applied the compost, it will be prevent from the leaching.
Do you think anything contributed from the composite for the improvement of the soil structure?
Yeah, it compost increase the microbial activity and others water holding capacity, so it will be improve the soil fertility and soil structure.
What type of soil did you use for your experiments and for this? Because different soils have different properties, of course, and also these properties determine how available these nutrients are to the plant. So I would like to know what kind of soil you used that you can use to say maybe it didn't leach much in this kind of soil, and in case of another soil, what do you think is going to be the effect on the plant?
Yeah, in Mediterranean desert the soil is loam, sandy loam maybe. I don't know about that.
Was there any what crop was growing on this?
First
in this field, and
when the sample was taken, the sunflower was grown there,
so
and after the harvest, then beige plant was.
So did you test the effect on the crop as well?
No, I just soil.
Other questions, please?
What
yes.
is the maximum, just compared to the literatures? What is the maximum amount of nutrient that you observe in the compost among the? Let's talk about the three main, nitrogen, phosphorus, and potassium. So, which one was the maximum compared to the recommended nutrients, which are commercially provided inorganically? Which one was the maximum that you got result?
The carbon. Carbon is carbon.
No, not about carbon. Just about the three primary nutrients.
NPK. Yeah, phosphorus. Yeah.
Okay, other questions to Fatta? So let us thank Fatta. We still have a few minutes for the next presentation, and there were some extra questions to Diriba that we were not able to discuss. So if you want to raise these questions, I just remind you we were speaking of hydraulic conductivity. Why laughing, Gabriel? Yes, Teklai. To Diriba, yes. Diriba, come here, please.
Okay.
I will bring back your presentation. We've finished the discussion. If you want, you can show.
Can I make a comment about Diriba?
Yes. Okay.
Okay. I was Diriba's mentor, so I just wanted to say that on the sap flow influence on sap flow, we were surprised to see that sap flow was reduced, and we don't really know why yet. And also it's a part of a much larger project in which many different aspects are being examined by different groups. So that all of the soil parameters and the salinity and all these properties are being followed. But Diriba was limited to just the sap flow, and he also measured some root properties, but he didn't have time to present, and they're still being analyzed. Okay, sorry.
Take like.
Okay, what do you think for another crops? It will be applicable for crops, not for trees.
Yeah, it varies based on the type of the crop, but we can use this secondary treated wastewater. Especially, there is a limitation for each crop, especially for those crops which can be consumed in fresh, especially vegetables. It is mostly not recommended to directly irrigate with this wastewater, as the contaminants may be used by the consumers directly instead of processed. But for the processed crops that we can further process, larger amount may be used, and less less attention will be given for fresh water, and we can use this secondary treated wastewater.
Let me ask you please a question to Diriba and maybe to Shabtai as well. Is the salinity, the electrical conductivity of the reclaimed water by itself not a sufficient reason to put the plant into some kind of stress and then cause it some stomatal closure or some other maybe aquaporins or whatever, and to slow down transpiration?
Can you answer? What was the salinity? You mentioned it.
I'll give you.
No, you first.
Okay, I think as we said, it may have an effect on the performance of the crop, as it may influence the availability of the pores in the plant, as we said, on the aquaporin. So it may have a direct effect, as I think, even though I didn't contact it and see it. I assume. It's my assume.
But when you're talking about salinity and we're talking about sap flow,
Okay.
you mentioned several times effect on aquaporin in terms of a factor of affecting sap flow.
Right.
Shuki mentioned correctly transpiration.
Mm-hmm.
So can salinity have an effect on transpiration as well?
Right, it does. During high transpiration, there may be high accumulation of this salinity. For example, sodium or calcium may be highly accumulated at the tip or at the margin of the leaves, that may cause the burning effect on the plant, and it may also cause the stomatal closure.
Okay,
Daniel?
Factor, just one comment. Okay, so what Diriba said, but he didn't say it again. The salinity of the fresh water was about 1 decisiemens per meter, and of the wastewater is about 2 decisiemens per meter. And 2 is really marginal for having a detrimental impact on citrus. Okay?
And also the short time.
And it's a short time, it's only a few months. That's why we were surprised. It's very good quality waste.
We are not speaking of detrimental effect. We're speaking of
Okay.
An effect.
minor effect.
Okay. It would be the first candidate.
Daniel?
I guess my question may be more for Dr. Cohen, but I just want to know how available is the secondary treated wastewater to farmers throughout Israel? Is it difficult for...
As he showed in the introduction, 50% of irrigation is with wastewater too.
Does it matter on location, or pretty much any farmer?
It's a matter of location, but in the south, the Shaftan is going to much of the central southern region, and in many areas, these places are located near reservoirs. Like the Mizra It has the wastewater reservoir for the whole Nazareth region. And that's, we're sitting right next to that reservoir.
The only region that is not allowed to use wastewater is the Upper Galilee, all the region that Magani Kavut.
The watershed.
The
watershed.
The watershed of the Kinneret. So just simply because they are afraid that some wastewater will reach the Lake of Galilee, the Kinneret. Other than this area, all over actually. Okay, Diriba, thank you very much, and thanks to everybody in the chapter for his contribution to this extra bonus discussion. And we shall move on to the last presenter of today, Ranjeet Shinde from India.
Good afternoon, all of you. I am Ranjeet Shinde, and the topic of my research is effects of NaCl salinity on onoriganum vulgare plants. This work was done under the supervision of Dr. Nidit Bernstein in Vulcani Center. First, I would like to give some basic background about my topic. What is NaCl salinity? NaCl salinity is the high concentration of NaCl salt in soil and water. About 23% of the world's cultivated land is saline. Thus, many crops are exposed to the salinity. In picture, we can see the NaCl salinity in desert. What are the effects of NaCl salinity on the plant growth? NaCl salinity reduces the growth of the root and shoot. In picture, we can see there is a reduction in growth of the roots and shoots with increasing salinity levels, and this reduction in the growth of root and shoot reduces the yield. We know what are the effects of NaCl salinity on the plant growth, but what is the mechanism of salinity damage? Firstly, salinity causes the specific ion effects. Under specific ion effects, we can include the toxicities and deficiencies. In toxicities, due to salinity, there will be the high concentration of the sodium and chloride in the soil. Thus, plant will accumulate the high amount of the sodium and chloride in them, and high amount of sodium and chloride is toxic to the plants. And in deficiencies, due to salinity, there will be the high concentration of sodium in the soil, and we know there is always competition between the sodium and other cations like calcium and potassium to get into the plants. And due to this, there will be a deficiency of such cations like calcium and potassium due to salinity in plants. Secondly, salinity causes the osmotic effects. Under osmotic effects, we can include the increase in solid concentration in tissues, and secondly, reduction in the water amount of the plants. There are three reasons for the increase in solid concentration of the plants. First reason is that under salinity, plant accumulate the high amount of sodium and chloride. Second reason, under salinity, plant absorb less amount of water. And third reason, for osmotic adjustment under salinity, plant produces the high amount of solutes. And also there are two reasons for the less amount of water in plants due to salinity. First reason is that due to salinity, there is a reduction in transpiration, thus plant take up less amount of water. And second reason, due to salinity, there will be the less difference of osmotic potential between the roots and the soil. And the difference between osmotic potential of the roots and soil is important for the uptake of water. Also, salinity is important for the oxidative damage. Due to salinity, plant produces reactive oxygen species, and this reactive oxygen species causes the oxidative damage. Example of oxidative damage is membrane leakage. In addition, in some cases, salinity is responsible for the sugar availability in the plant. There are three reasons for the less production of the carbohydrates in the plants under salinity. First reason, due to salinity, plant grow less, so there will be the less leaf area index. And second reason, due to salinity, salinity reduces the conductance of the stomata. And third reason is that due to salinity, there is a reduction in the photosynthetic pigments like chlorophyll A, chlorophyll B, and carotenoid. These three things reduces the accumulation of the carbohydrates in the plants due to salinity. We are studying the effects of salinity on Onoriganum vulgare plants. The origin of Onoriganum vulgare plant is Mediterranean region, and the family is Labiatae. In picture we can see the field of oregano in Peru. What are the uses of oregano? It is used as a fresh herb, dry herb, and also we can extract the essential oil from the oregano. In food science, oregano is used as a spice, and essential oil of oregano is used as a natural preservative. And in agriculture, essential oil of oregano is used as a bactericide, fungicide, and insecticide. And in medicine, essential oil of oregano is used as a stimulant and antiparasitic. Our hypothesis was salinity affects the water status and the mineral composition of the oregano plants, which jointly affect its growth and the essential oil production. Objectives. Our general objective was to study the effects of salinity on physiological responses and the essential oil production in the aromatic plant Onoriganum vulgare for evaluation of its suitability for the commercial cultivation under saline irrigation. Specific objectives of our studies were to study the salinity effects on plant development, plant ionomics, water relations in the plant tissues such as osmotic potential, cellular damage like membrane leakage, and essential oil production of the Onoriganum vulgare plants. What were the materials and methods used by us in this experiment? We grew plants hydroponically in 1/4 strength Hoagland solution, which was aerated. We used 6 salinity levels in this experiment: 1, 25, 50, 75, 100, and 125 millimolar. And 1 millimolar NaCl salinity level was the control treatment. For control treatment, we used 1 molar NaCl salinity level because NaCl is not essential element, but it's beneficial for the plant growth and development. We established our experiment in naturally ventilated greenhouse. Also, we used 5 replications per treatment with randomized block design. Before mineral analysis, we conducted 3 types of digestion and extraction. Sulfuric acid and hydrogen peroxide was used for the digestion of plant material for the extraction of nitrogen, phosphorus, potassium, and the sodium. While nitric acid and perchloric acid was used for the digestion of calcium and magnesium from the plant material. And we used chloride for the for chloride extraction. We used acetic acid. After digestion and extraction, we analyzed different minerals with the help of different instruments. Nitrogen and phosphorus was analyzed with auto analyzer, and potassium and sodium was analyzed by flame photometer. Atomic absorption method was used for the analysis of calcium and magnesium, and chloride analyzer was used for the analysis of chloride. Also, we measured different physiological parameters. Osmotic potential of the cell saps was measured with the help of osmometer. And membrane leakage. Membrane leakage is the sign of cellular damage. We measure membrane leakage in the form of ion leakage from the cells. Spectrophotometer was used for the measurement of photosynthetic pigments like chlorophyll A, chlorophyll B, and carotenoids after extraction with ethanol. Also, we extracted the essential oil from the plant materials. We extracted essential oil by hydrodistillation method. Results. First, results of plant growth. There was decrease in plant growth and development with increase in salinity level. Also, we measure the plant height, root length, number of nodes, dry biomass, and fresh biomass. Plant height was reduced with increase in salinity level, but there was no effect of salinity on the root length. Also, fresh biomass and dry biomass of the shoots was reduced with increase in salinity level, but there was no effect of salinity on the root biomass. This decrease in shoot biomass was due to the reduction in number of leaves and not due to the reduction in size of the leaves. This result suggests that the oregano is sensitive to the salinity, and shoot of oregano is more sensitive to the salinity than the roots. The results of ionomics: salinity induced toxicities. We observe there is accumulation of sodium and chloride due to salinity in leaves, stems and roots with increasing level of salinity, but the sodium and chloride concentration of the leaves and stems was higher than the roots, and the sodium levels in the leaves and stems are similar, but it's interesting to see that the stems accumulated higher level of the chloride than the roots. These results suggest that there was no reduction in the development and growth of the roots because of the less accumulation of the sodium and chloride in the roots, and reduction in growth and development of the shoots was due to the higher accumulation of the chloride in the stems. In the picture we can see the toxicities of NaCl due to salinity to the older leaves of oregano. Salinity induced deficiencies. We observe reduction in the potassium and calcium concentration of the leaves, stems and roots with increase in salinity level, but we did not observe any effect of salinity on magnesium concentration of the leaves, stems and roots. These results suggest that the reduction in growth and development of the oregano shoots was due to the reduction in the potassium and calcium concentration of the leaves, stems and roots. In picture we can see the calcium deficiency due to the salinity in younger leaves of the oregano. Also we analyzed other elements like nitrogen and phosphorus. We did not observe any effect of salinity on nitrogen concentration of the plants, but the phosphorus concentration of the roots was increased with increase in salinity level. Some previous studies shows that there is increase in phosphorus concentration of the plants with increase in salinity level, and some studies shows that there is decrease in phosphorus concentration of the plants with increase in salinity level. Also osmotic potential of the leaves, stems and roots was increased with increase in salinity level. Increase in osmotic potential of the plants under salinity is the part of mechanism of osmotic adjustment. Membrane leakage of the leaves and roots was increased with increase in salinity level. Increase in membrane leakage of the plants under salinity is the sign of cellular damage. This cellular damage may be due to the production of reactive oxygen species in plants due to salinity. Also we observed the decrease in chlorophyll A and chlorophyll B content with increase in salinity level, while the carotenoid content of the plants was increased with increase in salinity level. Increase in carotenoid content of the plants under salinity may be the part of mechanism of to prevent the oxidative damage. And also we observe one interesting thing here: chlorophyll B is more sensitive to the salinity in oregano than the chlorophyll A. Essential oil production of the oregano plants was decreased with increasing level of salinity. This decrease in essential oil production of the plants under salinity demonstrates the negative effects of salinity on plant metabolism. Conclusion: shoots of oregano are more sensitive to the salinity than roots. Reduction in growth of and development of shoots at higher salinity levels may be the results of reduction in leaf number. Toxicities of sodium and chloride and deficiencies of the potassium and calcium may be responsible for the shoot growth reduction. Increase in osmotic potential demonstrates osmotic adjustment under salinity. Increased membrane leakage under elevated salinity level demonstrates increased cell damage. Increase in carotenoid levels of plants exposed to salinity may be the part of mechanism to prevent oxidative damage. Decrease in essential oil production of oregano plants with increased salinity levels demonstrates negative effects of salinity on plant metabolism. suggestions for the next studies, further studies, to study relations between the salinity and fertilization, and to study the responses of oregano to salinity under soil cultivation. First, I would like to say thank you to Dr. Nirit Bernstein for her valuable guidance for me in my research. I am thankful to Dr. Netu Dudai for his help in experiment and for providing plants or seedlings for the experiment. Also, I am grateful to Rivka Rosenberg for her help in mineral analysis and data analysis. Also, I would like to say thank you to Avinam Smutrich for his help to set the experiment. And also, I am grateful to PEARS Foundation for providing scholarship for the research. Thank you for your attention.
Very nice. Okay, questions? Gabriel.
Ranjeet, you mentioned that once you are increasing the salinity levels, there is an increase. Obviously, there is an increase in sodium and chlorine in the leaves. So, and you also mentioned that there are certain properties or the uses for oregano as a spice, for example. So this changes these changes in the content of these elements, sodium and chlorine. Do you know if they change the taste or certain other?
Yes, definitely. They change the taste because the increasing sodium and chloride of salinity affects the essential oil production of the plants. That's why it changes the taste of the plants, because the taste of the leaves is mainly depends upon the essential oil. Yes.
Just a second, Tanmay.
I just said the salinity toxicity levels are more in older leaves.
Okay. Yes. Actually, generally, older leaves are more active than the younger leaves, and they are more transpiring than the younger leaves. So, due to the more transpiration, they accumulate higher amount of the sodium and chloride. So, there is a burning in older leaves, and also mainly we observe burning at the tip of the leaves, because like xylem vessels ends as the tip of the leaves. So, there is more accumulation of the sodium and chloride at the tips of the leaves. So, mainly we get the burning at the tips of leaves and not all over the leaf.
What influence do you think salinity has on the calcium deficiency and so on the graph?
Okay. Actually, salinity causes the deficiency of calcium in the plants. There are three reasons for the calcium deficiency. First reason is that we know always there is a competition between the sodium and cations like calcium and potassium. Thus, under salinity, there will be the high concentration of sodium in the soil. Thus, plants will absorb the less amount of the calcium. Second, we know the salinity reduces the transpiration, and transpiration is very important for the uptake of the calcium. And thus, the reduction in transpiration causes the reduction in uptake of the calcium. And also, root pressure is important for the uptake of calcium. Under salinity, we get the reduction in root pressure, because there will be the less difference between the osmotic potential of the roots and the osmotic potential of the soil. Thus, there will be the less uptake of the calcium. And mainly under salinity, we get the calcium deficiencies in young leaves or the fruits, because they are less active. And when they and there is a more need of the calcium, because calcium is important part of the cell. It's required for the cell oil development. And generally when they show the deficiency of any element, they act as a sink, and older leaves acts as a source. So there will be the flow of elements from the older leaves to the younger leaves or the fruits. But what happens in case of the calcium? Calcium is immobile in phloem. So it can't flow from the older leaves to the younger leaves or fruits. So mostly they shows the deficiency of the calcium in younger leaves or the fruits. Example of the calcium deficiency in tomato or pepper is blossom end rot.
So, next one, Shilpa, please.
Okay. You mentioned saying about you're measuring the things like ion exchange.
Okay.
And cellular damage. How are you going to measure those?
Cellular damage we measured in the membrane leakage.
How you're like, what is the method you're going to adopt there?
Okay, cellular damage. Yes, cellular damage we measured in the percent of ion leakage.
Ion leakage?
Yes, I'm explaining. First, when we harvested the plant, immediately we transferred the plant material to the distilled water. And for 24 hours we shaked it and we measured electric conductivity of the water. After that we autoclaved them for 48 hours.
You forgot to wash the leaves.
Yeah, we washed leaves.
I just asked that what is the method you're going to
I'm sure
I'm
sure he
That's
did
I'm
not
saying.
forget to do it in the lab. He just didn't.
I'm saying the method.
Yeah,
Wait.
but you didn't mention in presentation, I think, the electric conducting method. That's why I asked the question.
No, I told percent of ion leakage from the cells.
You said that leakage of cells from the tissues, but you
Okay.
didn't mention the electric conductivity method. You're going
Okay.
to use.
Mr. Wallin, please.
Ranjeet, I want to look at your conclusion once.
Okay. Sorry.
Where did you mention oxidation? Its relation to maybe here.
Yeah.
Increase in carotenoid number of plants exposed to salinity. Yeah. How
Yes.
did you conclude like this? Did you check the expression of some reactive oxygen species?
We didn't.
In relation to the ions, or how did you conclude this?
Can you explain, pardon?
He didn't say, just a second, he didn't say we measured, we found. He said that may be a part of. He did just expanding his.
No, I wanted to know if he read it from literatures.
Okay.
Okay.
Yo.
Ask your question again.
No, I just, if you know.
Why do you think that carotenoids may be involved in oxidative damage or prevention of oxidative damage? What's the connection between carotenoids and prevention of oxidative damage?
Okay, it's a good question. Under salinity, most of the times I read literatures. They explains that for oxidative under salinity or under any stress, there is a increase in carotenoid levels. Like this increase in carotenoid levels, it's maybe for the reduction of the damage of the salinity to the cells. Because under salinity also there is production of the reactive oxygen species, which damages to the cells. Thus, to compensate these things, plant produces the higher amount of carotenoid levels to get rid of the damages due to salinity to the cells.
Javiera.
Okay, I have two questions.
Okay.
One, do you know the role of the essential oil in the oregano? And if it has a role, how it will affect the decrease of them during salinity?
How will?
How will the salinity affect the essential oil?
Yeah, and the
Okay.
decrease of the essential oil, if they have a role, how they affect the performance of the plant. And the second question,
Okay.
I don't know, how many days did you grow it?
Yes, we grow plants for around 45 days.
Did you see any, just to know, flower induction?
No, they did not flower within 45 days. No.
Okay.
Yeah.
No, because I thought maybe.
So what about?
Yeah, flowering. Yes, it's very good question. Actually, flowering has influence on the essential oil production of the oregano plants. If there is a flowering, there is increase in the essential oil production of the some papers. I have read some papers from the Dr. Native Didai. He is from the Nemera. There is an increase in the essential oil concentration of the plants under flowering.
And what about the effect of salinity on essential oils?
It's different in different plants. Some plants like basil, there is an increase in essential oil concentration of the plants. But in oregano, we found the decrease in essential oil concentration of the plants under salinity.
Yeah, but sorry, you're not answering my question. What if you know the role of the essential
essential oil in the plant, and if it's decreased, if it has an effect on the performance of the plant?
Actually, it's a very good question, but there was a time limitation for the experiment, so we were not able to measure each and everything, so we didn't observe. Only we observed the production of the plants under salinity. What is the production of the essential oil under salinity? We didn't observe the what were the component of the essential oil or the what was the role of essential oil.
Okay, so I will take the last two questions.
Okay.
First of all, to what extent can you infer from your study to other species or maybe other cultivars of oregano? Is it like general phenomena what you measured, or it is very genotype specific?
I didn't got your question. What do you want to ask me?
The results you got.
Yes.
Now what cultivar was it?
It's Origanum vulgare.
Cultivar. Cultivar name.
I don't know.
You ask Nativ, he has name for any plant.
Okay.
So probably for this cultivar as well. Okay?
Okay.
So now I'm a farmer, I have a different cultivar. Can I learn from your results and assume that it will be the same on the other cultivar? Or if I'm not growing on oregano, I'm growing basil, zata, whatever, are these results also the same for the other crop?
Okay. For actually in case of salinity, many plants shows the different results for the salinity. For like in basil, like you will get the increase in essential oil concentration of the plants under salinity. But for oregano, we got the decrease in essential oil concentration. So it may differ from plants to plants or species to species. Also, actually we grew the plants za'atar, but there was a infection of diseases, so we were not able to test them.
Okay, now I have another very serious question. I mean, it's a question that really we were discussing it in my lab, and I'm not sure we got an answer. Maybe you have the answer. You see, when you grow plants under drought and you see a change in osmotic potential, this is simply just due to drought.
Okay.
But if under salinity you see a reduction, I mean more negative osmotic potential, namely higher concentration of solutes, it can be either the ions, the salt itself, or it can be accumulation of solutes, organic solutes by the plant as a means to cope with the stress. How can you distinguish? You've seen here a change in osmotic potential. How can you say increase in osmotic potential demonstrate osmotic adjustment? How can you say that it is osmotic adjustment and not simply just the poor guy got all these ions in there and here you get reduction in osmotic potential?
Okay, but because I told that it demonstrates the osmotic adjustment, because if there will be the if that was the poor guy, means that plant was the poor and it accumulates only salts, it's not due to the production of the solutes in the plants, then maybe it was died, but it was still alive. Means plants produced some solutes to compensate with the salinity for absorption of water, and it's still alive. Maybe it means the
well, I do
osmotic
agree.
adjustments.
I do agree that the increased osmotic potential, I call it decreased because it's becoming more negative.
Okay.
But nevertheless, we're speaking of the same thing. Okay. Increased osmotic potential, it does demonstrate some kind of change in the osmotic potential or adjustment. But how can I know if it is simply due to the salinity or real active response of the plant? Solutes.
Yes, because plant is still alive. If it is due to the salinity, plant will accumulate only salts.
This is not convincing me if it's alive, because many plants are alive and they do have salt in them. I mean, it's not that it went to far away, a very, very low osmotic potential. Nirit? Can
Well,
you suggest?
Yes, I think. I think part of what Ranjeet was saying is actually combined with what I'm going to say, is that we see accumulation on the figures and the results, accumulation of sodium and accumulation of chloride.
Okay.
But there's an ability to calculate what's the contribution of this amount toward osmotic adjust, toward the increase in.
If you know
osmotic-
this, the water content, and yeah,
Yes.
you can calculate.
And the extent of, normally, unless if you're talking about halophytes, then the increase in osmotic potential components due to sodium chloride probably cannot account for most of it, unless, again, depending on on changes in concentration of water. And how can we know? It's a good question, other than analyzing separately for organics and other sodium.
Yeah.
But also we should take into consideration, as you say, with the osmotic, and as Ranjeet also said in the beginning, that the changes in osmotic potential has also to do largely with changes in water. So it's reduction of water which also increase the concentration.
Okay. Thank you, Niri. Yes, please.
Okay, sorry, you said there's the last set questions, but I guess
You
lecture,
are
so
our honorable
I have
guest.
another question.
You get the right for the
I
last
wonder if
question.
this is the thing that there was less accumulation or less influence on the roots than on the stems and leaves. Is this a common observation in salinity? Salinity influences, that there's less of an influence on roots than on the stems and leaves?
Are you asking me, is it general phenomenon?
Yes.
No, it depends upon the plants. It depends upon the plants. Some plants accumulate the salinity or the sodium and chloride in the leaves. Some plants accumulate in the stem. Some accumulate in the roots. Or otherwise, some plants accumulate sodium in the roots and chloride in the stems. Some accumulate the sodium in the leaves, and some.
So then, what
depends upon the
ideas
plants.
would you have to explain why it's accumulating more in the stem, in the leaves, let's say, than in the roots?
Yes. Actually, it may be due to if the plant will accumulate the more sodium or chloride in the leaves or the younger leaves, so there will be no growth of the plants. And if the plant will acquire them in the leaves, in the older leaves or in the stems or in the roots, so plant can be growing to a certain extent. Not very well, but it can be grow up to a certain extent. Maybe it's because of this thing.
Okay, thanks.
Okay, so it's a time to thank Ranjeet first of all. And once again, to thank all the speakers of today for this very interesting day. And don't forget, we have tomorrow another day, not less interesting than this one. So, see you tomorrow morning.